Most people think blood vessels are just hollow tubes, simple plumbing. Blood goes in, blood comes out. But inside every blood vessel is a microscopic living layer almost no one talks about. A delicate gel-like porous coating inside the vessel wall. This structure helps regulate inflammation, oxygen delivery, clotting, vascular permeability, immune activity, and even blood flow itself. It's called the glycocalyx. And damage to the hidden layer may be one of the earliest events in atherosclerosis, diabetes, hypertension, and chronic inflammation. And here's what makes it fascinating. One of the molecules that may quietly help preserve this vascular interface is carbon dioxide. Not oxygen, carbon dioxide. And once you understand this relationship, you begin to see blood vessels in a completely different way. The glycocalyx is a thin, sugar-rich, negatively charged layer coating inside every healthy blood vessel. It sits directly on top of the endothelial cells. And rather than behaving like a static coating, it acts like a living biological interface. Imagine a dense underwater forest moving gently with the flow of blood. That's closer to the reality than smooth plastic tubes we often imagine. This layer is built from several key components. Glycoprotein, the structural backbone anchoring the layer to the endothelial surface. Proteoglycans act like molecular sponges holding water within the layer and giving it a gel-like consistency. Glycoaminoglycans are long sugar-based chains that carry a negative charge the glycocalyx depends on. And structured water fills the space between the molecules, organizing itself into an electrically active environment. Together, these components create a protective living environment between the blood and the vessel wall. The glycocalyx reduces friction, senses blood flow, regulates nitric oxide signaling, prevents excessive blood leakage, and limits immune cell adhesion. In a healthy vessel, red blood cells glide smoothly through hydrated, negatively charged environment. Flow becomes elegant, quiet, efficient. But when the glycocalyx breaks down, everything changes. One of the most fascinating things about the glycocalyx, it is highly negatively charged. And this matters profoundly because red blood cells are also negatively charged. Like charges repel each other. That repulsion keeps red blood cells spaced apart, reducing friction, aggregation, and turbulence. Instead of colliding, the cells glide. The glycocalyx also organizes water into a more structured state called exclusion zone water. Most of us think of water as a simple passive fluid, but near biological surfaces, water organizes itself into an ordered gel-like phase. It excludes particles and solutes, separates electrical charge, and begins to behave less like a liquid, more like a liquid crystal. Near the vessel wall, this structured water becomes more negatively charged, pushing protons outward into bulk fluid. What emerges is a spontaneous separation of charge across the vessel surface. The glycocalyx and its associated water begin to behave like a living battery, not metaphorically, electrically. Blood flow may be supported not only by the mechanical pumping of the heart, but by the electrical chemical organization of the vessel wall itself. And carbon dioxide plays a surprisingly important role in maintaining the conditions this system depends on. The glycocalyx is incredibly delicate, and modern life is almost perfectly designed to damage it. Chronic inflammation, oxidative stress, high blood sugar, smoking, disturbed blood flow, stress hormones, and chronically low carbon dioxide. When CO2 levels fall, blood vessels constrict, microvascular flow declines, oxidative stress rises, and the glycocalyx begins to shed. As this layer deteriorates, immune cells stick more easily, vascular permeability rises, fluid leaks into tissues, and endothelial dysfunction begins. The vessel wall becomes inflamed, sticky, and unstable. Before the artery clogs, the terrain collapses. And this may be one of the earliest stages of vascular disease. Carbon dioxide does not directly build the glycocalyx, but it helps preserve the environment the glycocalyx depends on. It does this through 5 interconnected mechanisms. First, CO2 improves microvascular flow. It relaxes small blood vessels, increasing perfusion through capillaries, and the vasovasvorum, the tiny vessels that feed the artery wall itself. When that flow declines, the artery wall becomes starved of oxygen and nutrients, making it far more vulnerable to change. And CO2 helps keep that flow alive. Second, the Bohr effect. As CO2 rises, hemoglobin releases oxygen more efficiently into the tissues. Paradoxically, carbon dioxide helps tissues receive oxygen not by increasing oxygen itself, but by improving where and how it is delivered. Third, CO2 reduces oxidative stress. Reactive oxygen species physically strip the glycocalyx from the endothelial surface. CO2 helps stabilize mitochondrial function, redox balance, and endothelial signaling, reducing that destructive burden. Fourth, CO2 helps maintain the proton gradients, an electrochemical environment that structured water depends on, sustaining the charge separation the glycocalyx relies on. And fifth, CO2 reduces excessive immune adhesion. It lowers inflammatory signaling and reduces the tendency for white blood cells to aggressively attach to the vessel wall. When CO2 levels are healthy, the glycocalyx remains hydrated, structured, negatively charged, and resilient. Blood flows more smoothly, oxygen delivery improves, inflammation becomes easier to resolve, and the vessel wall holds its integrity. When the glycocalyx is damaged, blood flow becomes chaotic. Microcirculation suffers, oxygen delivery worsens, and inflammation accelerates. Consider atherosclerosis. We often think of it as a cholesterol problem, but before plaque forms, the endothelial surface loses its protection. Immune cells adhere to the naked vessel wall. Inflammatory signals escalate, and the vessel becomes a far more hospitable environment for plaque to take hold. The glycocalyx may fail at first, the damage follows. The same pattern appears in diabetes. High blood sugar directly strips the glycocalyx, increasing permeability and accelerating endothelial dysfunction. This may explain why diabetic patients develop microvascular complications so consistently in the eyes, the kidneys, the nerves, and the heart. The terrain breaks down first, the damage follows. Maybe vascular disease is not simply a cholesterol problem. Maybe it's more of a terrain problem, a problem of microvascular flow, electrical organization, immune balance, and structural integrity. And maybe carbon dioxide has been misunderstood all along. Right now inside your body, blood is moving through an extraordinary living system. Not just vessels, not just fluid, but a charged, structured, intelligent interface between your blood and your vessel walls. And that interface is either being preserved or quietly broken down, moment by moment, based on conditions most of us never think about. Carbon dioxide is usually described as a waste gas, something to remove, something dangerous, but biology often reveals a deeper story. CO2 may be far more than a byproduct of metabolism. It is a regulator of blood flow, a stabilizer of structured water, a protector of mitochondrial balance, and a guardian of the glycocalyx itself. The health of your blood vessels may depend less on flow through them and more on the invisible living layer that protects them. Because before arteries fail, the glycocalyx fails. And by preserving the environment this structure depends on, carbon dioxide may help preserve one of the most important interfaces in the human body. Not just blood flow, but the terrain that makes blood flow possible.